Okay. So today we will be discussing antibody generation. Now antibodies are used in almost all laboratories, okay, for in vitro purposes. But antibody generation is a hectic process, right? Because in most of the cases we have read that antibodies are produced in the animal itself, right? But there are certain processes by which we can produce antibody outside of the animal body, okay. So today we will be discussing that. Now whenever we are talking about antibody, okay, you should understand one thing, that is there are basically two types of antibody. There are basically two types of antibodies. There are basically two types of antibody. And they are monoclonal, and polyclonal. Okay. So there are two types of antibodies, that is monoclonal and polyclonal. Okay. Now for the generation of monoclonal antibodies, we use what is called hybridoma technology. Okay. For polyclonal antibody generation, we inject a mice with a suitable antigen, then we get the polyclonal antibody. That means most of the natural antigens, most of the natural antibodies are polyclonal in nature. Okay. So right now obviously we will talk about hybridoma technology and monoclonal antibody formation. Okay. Now listen, you should not confuse these terms, hybridoma technology and monoclonal antibody formation, because hybridoma technology is actually a technique using which we can get monoclonal antibodies. You should understand this very carefully. Okay. Okay. Now how to get monoclonal antibodies in lab. Okay. Now listen, actually you cannot produce monoclonal antibody in lab why? Because monoclonal antibody formation or monoclonal antibody production is actually time consuming, number one. Number two, it is also heavy on your pocket. Okay. That means it requires much more, it is actually much more costly to produce a monoclonal antibody than to produce polyclonal ones. Okay. Now suppose you have got both the things, that is you have got time and you have got the money to produce monoclonal antibodies. Okay. So now you are ready to produce one. Now listen, how to produce monoclonal antibody? There are basic steps. Okay. Now listen, monoclonal antibody was actually produced at first by Kohler and Milstein. Okay. Now these two scientists at first showed the world that how to produce monoclonal antibodies. Okay. Now listen, before we start, we should know what is the difference between monoclonal antibody and polyclonal. Then you will understand the basic problem. Now listen, suppose this is a bacteria. Okay. Suppose this is the bacteria, and this bacteria have this type of antigens, suppose spikes, and suppose this is okay rectangle type of antigen. Okay. This is rectangle type of antigen. This is the bacteria. Okay. And this bacteria has one spike type of antigen and one rectangle type of antigen. Now listen, you should understand one thing. If there is a antibody which is designed like this, that is, this is the Fc portion, and this is the FAB portion. Okay. For more details about FC and FAB, go to the immunology lecture. Okay. Then you will understand it. Antibody structures. Okay. Now listen, this is the Fc portion. Now listen, In the FAB portion, you can see that this particular segment can bind with this, and this particular segment can bind with this. That means what I am saying is, actually this is a polyclonal antibody, okay, and in our body most of the times polyclonal antibodies are produced. Why? Because you should understand that, you know, there is something called, you know, in chemistry you have learned something called law of minimum. That means always in a reaction, that particular, you know, reactant is actually guiding the reaction mechanism which is used in the minimum concentration, right? In body also this principle is applied. Now listen, how? Suppose if there are two antibodies, like this, so now you can say this antibody will bind with this antigen and this antibody will bind with this, and this is suppose antigen number 1, this is suppose antigen number 2. So now you can see that the antigen number 1 will bind with this antigen and antigen number 2 will bind with this antigen, and they will attract macrophage, right, so that this whole bacteria can be eaten up. Now listen, the very basic thing you need to understand, why body instead of making these two antigens makes this type of antigen? Because in making these two antigens, the protein used will be double, but in making this antigen, the FC portion being common, the protein used will be minimum. That's why body in most of the cases produces polyclonal antibodies. But when we talk about specificity, listen with all your attention. When we talk about specificity, this term comes into antibody lecture, then polyclonal antibody is not suitable. Why? Because they can bind with both. There is no specificity for this particular antigen. Suppose this is A1, and this is A2, okay? So you should understand that this polyclonal antibody is not specific to A1 or A2, it is specific for both, that is, it is common for both. Now, whenever this specificity term comes into immunology lecture, then you should understand we are talking about monoclonal antibodies. Okay, MCAB. Now, okay. So this is the basic difference between polyclonal and monoclonal antibodies. Okay, we will talk about in details when we will actually finish this lecture. We will talk about in details the difference between monoclonal and polyclonal. Don't worry. Okay, we will make a chart. Okay, now listen. So monoclonal antibody generation was first shown by Kohler and Milstein. Okay, now what was the experimental design? You should understand this. Okay. What was the experimental design behind this? You should understand now that what was the experimental design behind monoclonal antibody generation. Okay, now listen. The experimental design behind Kohler and Milstein's monoclonal antibody generation was, you will have to take a mice. Okay, you will have to take a mice and you will have to inject the mice with, this is step 1, okay, learn every steps clearly. This is step 1. We'll have to take a mice, okay. Now there are many types of mice, bulb C, okay, mice or something like this. Okay, these are, you know, experimental mice. Now inject the mice with adjuvants first. Now understand this very basic thing. What is an adjuvant? Suppose this is an animal, and you introduce an antigen. Suppose this is an animal and you introduce an antigen into this animal's body. Now what will happen? Against this antigen, antibodies will be produced. right? Okay, let me draw it bigger. Suppose instead of this human, I am taking the example of a rat. Okay, now listen, suppose you introduced an antigen like this into the mice's body. Okay, now against this antigen antibodies will be produced, right? Right? What is an adjuvant? Now listen, what is the work of an adjuvant? Suppose in the second experiment you add same concentration of antigen. Suppose you add same concentration of antigen, okay, but to this antigen you are adding an adjuvant. To this antigen you are adding one adjuvant. What will happen is then, what will happen is there will be much more, production of antibody. That means an adjuvant increases the immunogenicity of an antigen. You should understand this. Okay? An adjuvant increases the immunogenicity of an immunology. What? Antigen. Now listen. You should understand one basic thing again, that there are many types of adjuvants which we can use, but the most common type of adjuvant, okay, the most common type of adjuvant we use is Freund's adjuvant. Okay, now if you search it, what is a Freund's adjuvant, you will get to know something like this, that Freund's adjuvant has two basic components, that is incomplete and complete Freund's adjuvant. Okay, now not going into the details further, you should understand that when we will be injecting these mice with adjuvant, you should understand we are injecting these mice with Freund's Freund's complete plus Freund's incomplete adjuvant. Incomplete adjuvant. Okay, you should understand this. That means you need to understand that at first, for the preparation of monoclonal antibody, you are injecting this mice with Freund's complete plus Freund's incomplete adjuvant. Okay, now what will happen is, actually